Hi, I am Ancy, Department of Biomedical Engineering from Emmanuel Sir, JG College of Engineering. Today we are going to discuss about biosensor. What is sensor? What is biosensor? Biosensor is an self-contained integrated device that is capable of providing specific qualitative, semi-qualitative and semi-quantitative analytic information using a biological recognition element. What is the biological recognition elements? Which is direct spatial contact with a transduction element. In the biosensor, the current definition of the biosensor is a sensor that integrates a biological element with a physiochemical transducer to produce an electronic signal proportional to a single analytic, which is then conveyed to a detector. In the biosensor, already I told what is biosensor. Biosensor is an analytical device used for the detection of an analyte that combines a biological component with a physiochemical detector. The biosensor device, which there is a coupling of biosensing element with a detector system using a transducer in comparison with any other currently available diagnostic device. The biosensor must having a higher performance in terms of the biosensors are the sensitivity and selectivity both. It has both sensitivity and selectivities are the higher in the biosensor. The biosensors have found potential application in the industrial processing and monitoring, environmental pollution control, also in agriculture and food industries. The important features for commercialization of the biosensors are selectivity, sensitivity, stability, reproducibility and the low cost. What are the features of the biosensors? The features of the biosensors are selectivity, sensitivity, stability, reproducibility and low cost. In the biosensors, mainly the biosensors introduced Leland C. Clark. He is invented the biosensor, that is called Clark oxygen electrode, a pivotal device that allows real-time monitoring of patient blood oxygen levels and has made surgery safer and more successful for millions around the world. The father of biosensors called Leland C. Clark. He introduced a real-time monitoring device that is blood oxygen levels and made surgery safer and more successfully for millions around the world. What are the components of the biosensors are? What are the components of the biosensors are? Analyte, bioreceptor, transducer and measurable signal. Analyte means some biological constituents having the analyte. The bioreceptor having enzyme, antibody, microorganism. These microorganisms are filtered and absorbed transducer. In the bioreceptor, electroactive substances. The transducer should transduce the pH change, heat, light, mass change. Then the bioreceptor having the piezoelectric device, photon counter, thermistor, semiconductor pH electrode and electrode. These are the inside the transducer. The transducer should be used in the signal transducer. The signal transducer contain the electrode, semiconductor pH electrode, thermistor, photon counter and piezoelectric device. Then finally measurable signal. The measurable signal they converted into electric signal and detect the biosensors. The biosensors are mainly divided into three sections. First one is sensors, second one is transducer, third one is detector. The sensors having the biological material, that is tissue, microorganism, organelles and cell receptors, enzymes, antibodies, nucleic acid. These are the sensitive biological elements. The sensitive biological elements are the tissue, microorganism, organelles, cell receptors, enzymes, antibodies and nucleic acids. Then next one is transducer. The transducer is the detector element that works in the physiochemical way, optical, piezoelectric and electrochemical, that transforms the signal resulting from the interaction of the analytic with the biological responsible for the display of the result in a user-friendly way. Third section is the associated with the electronics, which comprises of signal conditioning circuit. What is the signal conditioning circuit? The signal conditioning circuit acts as the amplifier. What is amplifier? Amplifier is an amplify the signal, processor and the display unit. The biosensors, the elements of biosensors, the biosensors having the bio element and sensor element. What are the bio element? The bio elements are the already told enzyme, antibody, nucleic acid, tissue, microbial and polysaccharide. What are the sensor element? The sensor elements are electric potential, electric current, electric conductance, electric impedance, intensity and phase of EM radiation, mass, temperature and viscosity. The biosensors mainly classified into two elements, physical elements and biological elements. What are the physical elements? Physical elements are called transducer and amplifier. Biological elements are the sensitive bioelement and analytic. The principle of biosensors. The biosensors having the membrane envelopment. The membrane envelopment having the non-covalent or covalent band. This immobilized biological material is in intimate contact with the transducer. The analytic bind to the biological material to form a bound analyte, which in turn produce the electronic response that can be measured. In some instances, the analytic is converted to a product which may be associated with the release of heat, gas, electrons or hydrogen ions. The biosensors is converted to a product may be associated with release of some gases, heat, electrons and hydrogen ions. The transducer can convert the product linked changes into electrical signals which can be amplified and measured. The biosensors having the physical element and biological element. These elements are having the sensors, transducer and detectors. The sensors are sensing the element. Transducer may convert the product linked changes into electrical signals which can be amplified and measured. The measurement flow of biosensor. The measurement flow of biosensors are biomaterial and analyte combined with bound analyte, biological response, electronic response and measurement working of a biosensor. The electrical signals from the transducer is often low and superimposed upon a relatively high and noisy. The biosensor having the electrical signal. These electrical signals having some high amount of noise. They contain what that means, that contain high frequency signal component of an apparently random nature due to electrical interference are generated within the electronic components of the transducer. The signal processing normally involves subtracting a reference baseline signal derived from a similar transducer without any biocatalyzed membrane from the sample signal, amplifying the resultant signal difference and electronically filtering problem of electrical noise filtration. The biosensors in such cases having the analog signal produced at the stage by the output directly, but is usually converted to a digital signal and passed to a microprocessor stage where the data is processed, manipulated to desired units and output to a display device of data. The most of the biosensors is filtering. Filtering means smoothing the unwanted signal noise. The biosensors having the unwanted signals, these signals are filtered by using the filtering. It has been the catalyzed membrane, sampling the signal, amplifying and electronically filtering. Then the biosense technology. Nowadays, the biosense technology may grow mainly through the generations of the three generations of biosense mainly explain. The first generations of biosensors were the normal product of a reaction diffuses to the transducer and cause electrical response. In the sense, the first generation of biosensors, it is a normal product of reaction diffuse to the transducer and cause the electrical response. The second generation of biosensors act as the mediators. It is having the mediator between the reaction and the transducer in order to generate improved response. And the third generation biosensors where the reaction itself causes the response and no product or mediator diffusion is directly involved. This technology, detection and recognition. First one is the antibody having the some enzyme, then cell membrane receptors, then competitive binding. The competitive binding have polymer and hydrogel. Then second generation have the signaling principles. Signaling principles having the signaling principles having the optical, electrical. The electrical mainly consists of voltammetry, potentiometry and conductivity. Then signaling principles having the electromechanical, then thermal, magnetic and pressure. The biological molecule may immobilization permit repeated use of costly. Then the biosensors. The biosensors explained it is an magnetoelectric biosensors, mainly transducing element and recognition element. The transducing elements mainly classified into mass-based biosensor, electrochemical biosensor and optical biosensor. The mass-based biosensor. What is mass-based biosensor? The mass-based biosensor consists of magnetoelectric biosensor and piezoelectric biosensor. Mainly the piezoelectrics. What are the piezoelectrics? Piezoelectrics are the quartz crystal microbalance. That means QCM. Then surface acoustic wave. That is SAW. The mass-based biosensor classified into magnetoelectric, piezoelectric. The piezoelectric again classified into quartz crystal microbalance, surface acoustic wave. Then the again transfusing element may classified into two. First one is electrochemical biosensor and the optical sensor. The electrochemical sensors having the potentiometric sensor, amperometric sensor, impedometric sensor, conductometric sensor. These sensors are the sensing the ampere, impedance, conductivity and potentiometric of the electrochemical biosensor. Then optical biosensor. The optical biosensors having the what are the optics? Opticals means it is having the fibers, fiber optics and FDR and surface plasma resonance, that means SPR. Then next one is biorecognition element. Already we know what are the bioelements having antibodies, DNA, enzymes. These are the bioelements. The biosensors. The biosensors mainly used piezoelectric sensors. Piezoelectric biosensors are the mass-based biosensor. Piezoelectric biosensors are based on the principle of acoustic. What is acoustic? Acoustic means sound waves. Acoustic, the principle of acoustic means the biosensors absorb the acoustic and receive the vibration. Hence, they are also called as acoustic biosensors. Piezoelectric biosensor produce an electrical signals when a mechanical force is applied. Piezoelectric produce electric signal then when a mechanical force is applied. In this mode, sensing molecules are attached to a piezoelectric sensor. The biosensors having the piezoelectric sensor, the piezoelectric sensor having the some modes. These modes are the sensing molecules are attached to a piezoelectric surface. In this piezoelectric surface, attached the sensing molecules. These sensing molecules a mass to frequency transducer in which interaction between the analytic and the sensing molecules set up mechanical vibration that can be translated into an electrical signal proportional to the amount of analyte. What are the analyte? Analyte having these some microorganisms, microbes and some DNAs. This is the example of piezoelectric sensors is quartz crystal micro and nano balance. In the biosensors mainly classified into already we know piezoelectric sensor, electrochemical sensors and optical sensor. They are already explained. Piezoelectric sensors are already know. Piezoelectric sensors is the quartz crystal micro or nano balance. They having the biosensors. It is a mass based biosensor. Biosensors, piezoelectric biosensors are principle of acoustic. Acoustic means sound vibrations. Piezoelectric biosensors produce an electric signal when a mechanical force is applied. Then electrochemical sensors. Electrochemical sensors are the have an subject of basic as well as applied research in nearly 50 years. Nearly 50 years, electrochemical sensors have applied research. In the already I told the biosensors are invented Leland C. Clark. Then again Leland C. Clark introduced the principle of first enzyme electrode with immobilized glucose oxidase at the New York Academy of Science Symposium in 1962. In Science Symposium in 1962, Leland C. Clark introduced the principle of first enzyme electrode with immobilized glucose oxidase. Then in this configuration, sensing molecules are either coated onto or covalent bonded to a probe surface. A membrane holds the sensing molecules in place, excluding interfering species from the analytic solution. The sensing molecules react specifically with the compounds to be detected. Sparkling, the electrical signal proportional to the concentration of the analytic. The concentration of analytic means already we know what is the analytic microorganism, DNA, enzymes, these are the analytic. Based on their operation principle, the electrochemical biosensors can employ potentiometric, amperometric and impediometric transducer, converting the chemical information into a measurable amperometric signal. The electrochemical sensors having the operating principles such as potentiometric, amperometric and impediometric transducer. It has converting into the chemical information into a measurable amperometric signal. Then optical sensors. Optical sensors, we know optical means it's having the fiber optic sensors. It is optical fibers allow detection of analytes on the basis of absorption, fluorescence or light scattering. Here both catalytic and affinity reaction can be measured. The reaction causes a change in fluorescence or absorptions resulting due to change in the refractive index of the surface between two media which differ in density. For instance, if bodies bind on a metal layer, the refractive index of the medium in contact with this layer will change. Since they are non-electrical optical biosensors having the advantages of lending themselves to an vivo application and multiple analytes to be detected by using different monitoring wavelength. The optical sensors basis of absorption, fluorescence and light scattering. It is the non-electrical biosensors having the advantages. It is applicable to be detected by using different monitoring wavelength. The versatility of fiber optic probes is due to the capacity to transmit signals that reports on changes in wavelength, wave propagation, time intensity, distribution of the spectrum or polarity of the light. The optical sensors have changes in wavelength, wave propagation, time, intensity, distribution of the spectrum or polarity of the light. These are the main things of the biochemical sensors. The biosensors used in various fields. First one is clinical and diagnostic application. In the diagnostic application, the biosensors are mainly used. Then industrial application, then environmental monitoring, agricultural monitoring and food industry. The basic characteristics of the biosensors are linearity, sensitivity, selectivity and response time. Linearity should be high for the detection of high substrate concentration. Then sensitivity, value of electrode response per substrate concentration, correct substrate concentration. Selectivity means chemical interference must be minimized for obtaining correct result. Then response time necessary for having 95% of the response. What are the basic characteristics of biosensor? Linearity, sensitivity, selectivity and response time. The advantages of biosensors. What are the advantages of biosensors? Biosensors are highly specific. It is rapid, accurate, stable and sterilizable. Another advantage is independent of factors like stirring pH. It is a linear response and tiny biocompatible. It is an easy to use, durable, require only small sample volume. What are the advantages of biosensors? It is an highly specific, independent of factors like stirring pH, requires only small sample volume, rapid, accurate, stable and sterilizable, easy to use and durable, linear response and tiny biocompatible. In the biosensor, same as like biosensor, some typical sensing techniques having. What are the typical sensing techniques of biosensors? First one is QCM. Already we know QCM is quartz crystal microbalance. QCM are the one of the sensing techniques. Then SPM. SPM in the sense scanning probe microscope. Then impedance spectroscope, DNA microarray, fluorescence, electrochemical, then SERS. What are SERS? SERS is the surface enhanced Raman spectroscopy. Then SPR. What is SPR? SPR is surface plasma resistance. Then some types of biosensors. This biosensors, what are the types? Calorimetric and thermal reduction biosensor, optical biosensor, resonant biosensor, piezoelectric, ion sensitive, electrochemical biosensor. We already know these are the overview of the biosensor. What are the types? Then sensing techniques, advantages, basic characteristics. These are the biosensors overview. The electrochemical biosensor again classified conductometric, amperometric, potentiometric. The biosensors for agriculture and food industry. Detection of viral, fungal, bacterial disease of plants in food industry. Detection of total microbes and food quantification in soft drinks to determine the freshness of other fish, beef and other food items. Makes bacteria flow by optical biosensors. Then we are going to discuss pH measurement. What is pH? pH is the number of instance how acidic and basic a solution is. A low pH means high acidity, a high pH means high alkalinity, which pH is 7 is neutral. The pH measurement, the pH is the base minus log, the base 10 logarithmic H plus. The ionic concentration, the mainly measured. The factors affecting the pH. Several factors influence the pH of a solution. One is concentration of acid or base. More concentrated acid and base yield higher H plus or OH minus. That means more concentration acid yield higher H plus and base are the explained OH minus. In the pH temperature affect ionization constants. For example, pure water have pH of 6.14 at temperature 50 degree, not 7. In the pH, presence of buffers. Buffers resist pH changes when small amount of acid or base are added. Ionic strength affects activity coefficient, especially in concentrated solution. Then chemical composition, polyprotic acid or amphioprotic substances can alter the overall ion balance. Then pH scale. pH scale ranges from 0 to 14. Under standard condition, temperature 25 degrees centigrade, though values extend slightly beyond this range in concentrated or non-aqueous solution. pH below 77 acidic solution, equal to 7 neutral solution. The pure water is an pH 7, greater than 7 basic, that is alkaline. Example, battery acid pH 0, lemon juice pH 2, pure water pH 7, baking soda solution pH 9, bleach, that means by using dress wash powder. Importance and application. Vital role in real world context. Maintaining blood pH, adjusting soil condition for crops, producing safe and effective medication, fundamental control parameter across science and industry. The pH application mainly used in science and industry. Main role in real world context. Biology. In biological field, enzyme activity, cellular respiration and homeostasis depending on optimal pH range. Medicine. Blood pH is 7.35 to 7.45 is tightly regulated. Acidos and alkalis are serious disorder. In agriculture use, soil pH affect nutrient availability of crop yield. Then environment, ocean acidification and freshwater pH impact ecosystem. Food science, fermentation, cell life and flavor depend on pH. Food science. Industry, pH control is essential in water treatment, cosmetic, pharmaceutical and chemical manufacturing. Then PO2. PO2 measurement is a partial pressure of oxygen dissolved in blood reflecting lung oxygen uptake and oxygen available for time. PO2 means partial pressure of oxygen. How many amount of oxygen available in tissue, that mean measured. PO2 or partial pressure oxygen represents amount of oxygen gas dissolved in a liquid such as blood. This measurement provides insight into how effectively oxygen moves from the lungs into the bloodstream. Understanding PO2 is important for assessing respiratory health and body's ability to acquire and utilize oxygen. What is PO2? PO2 means partial oxygen. It is used to measure the how many amount of oxygen present in the oxygen gas dissolved in a liquid such as blood. PO2 measurement, partial pressure of oxygen in blood measured by O2 electrode to determine oxygen content. PO2 electrode means Clark electrode, measures the current that flows and constant voltage is applied to the system. As dissolved O2 diffuse from the blood, a change in current occurs which of PO2 measurement. The PO2 mainly value is 13.3 Pascal. Then PCO2 measurement. PCO2 means partial pressure of carbon dioxide, reflecting lung ventilation and acid-base balance. Measurement method, atrial blood gas analysis. The most common method is atrial blood gas testing. Small sample of arterial blood is drawn, usually from the wrist, and analyzed pH, oxygen and PCO2. Mainly the PCO2 measurement method, atrial blood gas analysis, which are called ABC analysis. The atrial blood gas testing, small sample and atrial blood is where did we get from the wrist and analyze for pH, oxygen and PCO2. The method directly measures the dissolved CO2 gas, the blood and provides accurate information about respiratory function and acid base status. The next one is transcutaneous CO2 monitoring. A non-invasive alternative is transcutaneous CO2. Transcutaneous means it's on skin surface. The electrode should be placed on the skin surface and measure the PCO2. In this method, use continuous monitoring, especially in patients with sleep. When the patient sleep, that time the CO2 monitoring. Breathing disorders and pediatric and critical care settings by using transcutaneous CO2 monitoring. The transcutaneous CO2 monitoring mainly used for breathing disorders or in pediatric and critical care settings. Then high PCO2. High PCO2, that means greater than 45 mmHg. It indicates hyperventilation or respiratory acidosis, which may occur in conditions like chronic obstructive pulmonary disease, that means COPD, or respiratory failure. The respiratory failure, high PCO2 occur in the respiratory failure greater than 45 mm Hg.